KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Hi students, now in this video we are discussing remaining cell organelles of endomembrane system. Okay, so remaining cell organelles of endomembrane system. So now in a endomembrane system totally how many number of cell organelles present we have learned in previous video. In an endomembrane system totally 4 different types of cell organelles are present, isn't it? So first one we have discussed, that is endoplasmic reticulum is seen. After endoplasmic reticulum, now so 3 are remaining. So these 3 videos in this video, so 3 cell organelles now, so we are discussing in this video. Now second one is Golgi Apparatus. Now Camillo Golgi in 1898. Camillo Golgi. So who discovered this particular Golgi apparatus of first time means Camillo Golgi in 1898. Okay, so in 1898 so this particular person discovered. Now here, so what is the main important point now here we are discussing is Camillo Golgi in 1898 first observed densely stained reticular structures. Densely stained, one more time once you see. So first this particular person Camillo Golgi observed or noticed that densely stained reticular structures nearer the nucleus. Means what are the structures or densely stained reticular structures which are present nearer or which are present very near of nucleus. No, so all those structures he has named it as or he was named it as Golgi Apparatus. So now later, what is the point? No, so they were later named it as Golgi bodies after him. Okay, so after that particular scientist, no, these particular reticular structures only now we will be calling as Golgi bodies. Okay, so why we are calling Golgi bodies? Because of the name of Camillo Golgi. So that is the point we are learning now. Next one, they consist of many flat and disc shaped sacs. One more time, they consist of many flat and disc shaped sacs or cisternae of 0.5 micrometers to 0.1 micrometer in diameter. So here what is the point here we are discussing is, so these Golgi bodies they may consist of or they consist of many flat and disc shaped sacs. Okay, suppose if you see there once if you are looking on this particular structures, now here totally 3 different types of structures are there. Okay, so here vesicles are there, cisternae is there and tubular structures. So here one tubule like a structure is there, no, so with white color, so that one. Likewise there are many flat and disc shaped sacs or cisternae. So it is cisternae. So here this particular orange color structure is there, no, or cisternae, and the diameter of this particular cisternae is 0.5. Here point is there 0.5 micrometer to 0.1 micrometer in diameter. Next one, these are stacked parallel to each other. So these are all like bunch mother. So one by one, so arranged each other and stock like structure and parallelly arranged. So these kind of structures are arranged parallelly. So all these are all structures arranged parallel to each other. Next one, varied number of cisternae are present. So varied number means there are different types of cisternae are present in Golgi apparatus near the nucleus. So with the distinct convex cysts or forming phase. So here this particular Golgi complex, if you see there, so that particular Golgi complex we can be divided into two parts. So here two sides, not parts, sides. So one side we will be taking as a cis region or forming phase, and another region we will be taking as a concave, trans or maturing phase. So once you see here in a diagram, once I will show you, sir, where is a cis region or where we have to take a cis region, and on which side we have to take a trans region, means once you see the entire diagram of Golgi apparatus, so upside here, in this side, exactly at spotlight moving now. So that side you try to take it as a cis region, and here the spotlight is moving now. Here we can be taken as a trans region. Means once you see again, here the cis region we can take, and here the trans region we can take. Okay, so thereby, so what are the, sir, why, sir, here what is the use of this cis region and trans region, why we are learning about this one means, so definitely in a few minutes later you will learn that one. Now the cis and trans phases of the cellular organelle or the organelle are entirely different. So here the cis regions as well as the trans regions, definitely if you see there, definitely differences are there. So what is the importance of those phases? One is forming phase is there, and one more is maturation phase is there. What are the uses of this forming one and maturation? So very soon you learn. Next one, once you see there, next one, but so here these all means here the cis region or the trans regions or seems to be very different, but all are but these are interconnected. So means here even though these cis region and trans region are structurally different, but all these two structures are interconnected. Next one, Golgi apparatus, if you see there, principally performs the function of packaging of materials and materials to be delivered either to the intracellular targets or secreted outside of the body or outside of the cell. What is the point now we are learning? So principal function, don't forget, it is even a NEET examination point of view also important in competitive examinations. Once you see, Golgi apparatus principally performs the function of packaging of materials. Definitely, what is the main important function of Golgi apparatus means, it performs as a how functioning means like a packaging of materials, like a delivery, like a Flipkart or Amazon likewise. So some kind of packaging of materials to be packed and delivered. Okay, means here the what are the main materials which are present in this cell? All these materials are packed. And what are the materials are packed in the form of vesicles? All these are all will be transported or delivered once you see delivered either to the intracellular targets. Intracellular, intra means within the cell, so that may transport or deliver to the intracellular targets, or that may secreted outside of the body, means intercellular. Point you understood, means thereby. So here two important functions of this particular Golgi complex we are observing. So what are those? First of all, it pack. What it will pack? It packs the food materials, or it packs the materials of food vendor. So it packs the materials, and that definitely after packing of that particular materials, that deliver into the intracellular targets or secreted outside of the cells. Point you understood. I hope you understood this one. Now, materials to be packed in the form of vesicles. So here, what are the materials here we have studied, packaging of materials. So now author giving a name. So that particular materials or the packed materials name is vesicles. Once you see here, materials to be packed in the form of vesicles. So, sir, vesicles means what, sir, means once you see in a diagram, chinna motor like structures, spherical zero is there, so this kind of structures only our subject point of view related to the Golgi apparatus we are calling as vesicles. point you understood ipo parengama what is the point now I am explaining no principal function of Golgi apparatus packaging of materials in the form of vesicles. So now here from the endoplasmic reticulum fuse with the cis phase of the Golgi complex and move towards the maturing phase. So means here once you see there this continuous line one more time you read there materials to be packed in the form of vesicles from the endoplasmic reticulum fuse with the cis phase of the Golgi complex means so this one is a white chalk piece with a wet chalk piece we have drawn now adi nuclear membrane with pores. Next one pink color is there now that one is endoplasmic reticulum. Ipu endoplasmic reticulum nalli idi cis region. So here I have marked a cis region and this one is a trans region, isn't it? So there, so this is the structure of Golgi apparatus. Once you see there, this one is a nuclear membrane is there, endoplasmic reticulum, and here this one is a cis region and this side is a trans region. So likewise, so here this particular cis region of the Golgi complex is situated very close to the endoplasmic reticulum. Means here all these materials are packed in the form of vesicles. So what are the materials are there? All these materials packed in the form of vesicles. So here definitely what is the point here we can discuss is, so what are the materials are there? So all these materials are packed in the form of vesicles. So next one here, so once the materials is packed in the form of vesicles, okay ma, now here what is the point? What are the vesicles are there? All these vesicles, so definitely formed from the so packed from the endoplasmic reticulum, so which is fused with the cis phase of the Golgi complex. So once you see here, so exactly at spotlight we can see the packaging of materials. So means that is very close to the endoplasmic reticulum with the cis region of Golgi complex. Point you understood means that region is related to the cis region of Golgi complex only, but very near of endoplasmic reticulum. That's why here once you see materials to be packed in the form of vesicles from the endoplasmic reticulum fuse with the cis phase of Golgi complex and move towards the maturing phase. So definitely here in packaging of materials point of view, definitely endoplasmic reticulum and the cis phase of Golgi complex involvement is there. And what are the materials are packed in the cis region of Golgi complex? Definitely move towards the maturing phase. Definitely move towards the maturing phase. Next one, this explains. So thereby what is the point here we can understand? No, this explains. So this one explains why Golgi apparatus remains in close association with endoplasmic reticulum. So thereby we can understand, so in packaging of materials in the form of vesicles, so definitely, so here Golgi apparatus as well as endoplasmic reticulum both must be coordinated. Why? Because so these all four cell organelles functions are coordinated now. So because when these functions are coordinated, that's why these four cell organelles we have been included under endomembrane system, isn't it? So thereby what is my point is materials packaging everything taking place in a cis region of Golgi complex and moving towards the maturing phase. Cis region of Golgi complex means here we learn now forming phase, and a concave region or a trans region we are calling by one more name, maturing phase. So thereby what are the materials are prepared in are formed in the forming phase, so maturation process taking place in maturing phase or delivered towards the maturing phase or through the maturing phase. Next one a number of proteins. So definitely, so thereby here what is the point? This explains why Golgi apparatus remains associated with the endoplasmic reticulum. So thereby we can understand definitely endoplasmic reticulum and Golgi complex both must be associated. Okay, why? Because here packaging of materials everything we have seen. Next one, a number of proteins. This particular point also important. A number of proteins synthesized by ribosomes. ఇప్పుడు again here ribosomes where we can find out on endoplasmic reticulum. ఇప్పుడు a number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi complex before they released from its transing phase. Transphase. So thereby, so materials or the proteins, proteins definitely which are synthesized in endoplasmic reticulum. Why? Because in endoplasmic reticulum, because of the presence of ribosomes on the endoplasmic reticulum, rough endoplasmic reticulum formation takes place. That particular rough endoplasmic reticulum involves in the formation of it helps in the synthesis of proteins. So thereby, what are the proteins are synthesized in a rough endoplasmic reticulum? No, all these proteins are come like that. So all these proteins definitely come through the cisternae region of or cisternae of the Golgi complex. Means in a cisternae of the Golgi complex all these proteins are modified, and after what are the proteins are modified in a cisternae region? No, all these kind of proteins are released from the transphase of the Golgi complex. So once you see here diagram is there, no? So here protein synthesis process taking place. So here what are the proteins are synthesized in a rough endoplasmic reticulum? No, all these are all moved like this. So first of all it will come through the cis region, and in a cis region, no, cisternae region is there, no, cisternae, in a cisternae all proteins are modified and released that side towards the trans region. So likewise main important points of this particular Golgi complex we can see. Next one, Golgi apparatus is the important site for the formation of glycoproteins and glycolipids. Golgi complex definitely involves in the formation or involves, or it is important site for the formation of glycoproteins and glycolipids. Glycoproteins and glycolipids, ఇది NEET examination point of view important. Okay, so definitely Golgi apparatus helps in the formation of, or it is acting as important site for the formation of glycolipids as well as glycoproteins. So this is a rough diagram of which one? Golgi complex or Golgi apparatus. Next one lysosomes paranga. So this particular diagrammatical explanation kurtachi already. Now here the lysosomes information. So these are membrane bound vesicular structures formed by the process of packaging of materials in the Golgi apparatus. So thereby here what is the point we are discussing? What are the vesicles? What are the vesicles or the packaging of materials we see? No. So principal function of Golgi complex is packaging of materials in the form of vesicles. We don't know. Those vesicles only we are calling as lysosomes. Vesicles are nothing but, so here in a diagrammatically we have seen. So here chinna motta structures are there, no zero like structures, so spherical shaped structures. All these are spherical shaped structures only now we are calling as lysosomes. Okay, now the isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes. Very important point. Means if you see these particular structures, means what are the vesicles which are packed in the form of what are the particles which are formed in the or which are packed in the form of vesicles? No, definitely these vesicles consist of very rich almost all types of hydrolytic enzymes. I mean, so there definitely, so we can see the presence of hydrolytic enzymes. For example, hydrolases, there are different types of enzymes: lipase, protease, carbohydrates, and etc. Or etc. Vendor, these three only try to remember one more time. So what are those hydrolytic enzymes? Means lipase ends with ASE. Suppose if any term or word ends with ASE, stands for enzymes. So now lipase, protease, carbohydrates. So three names or three words names also ends with ASE only, you see there. So stands for enzymes. So there, so optimally active, so means here these are hydrolytic enzymes are optimally active at acidic pH. So all these kind of hydrolytic enzymes are optimally active at acidic pH. This is neat point of view important. Next one, these enzymes are capable of digesting of lipids, proteins and carbohydrates. So thereby how no lipids or the lipids can be digested by lipase. Next one, protease digest the proteins. Next one, carbohydrates are digested by the carbohydrates. But here what is the main important point we have to remember, no? What are the hydrolytic enzymes are there, no? All these hydrolytic enzymes are very active at acidic pH. Acidic pH means I think so up to 6.5. Why? Because 7 is a neutral, no? So that's why. So below 7, so we can be taken as acidic pH, isn't it? So there. So small information only related to the lysosomes. Okay, lysosomes are nothing but, so what are the materials packed in Golgi apparatus, no? So such kind of packed materials only we are taking as lysosomes. So rich with hydrolytic enzymes, so for example lipase, protease, carbohydrates, so will be very active in acidic pH. Definitely digest the lipids, proteins and carbohydrates. Idu podu. Next, next cell organelle and last cell organelle in endomembrane system. So last cell organelle is vacuoles. So here 1 or 2 points are there to be discussed. Next one, the vacuole is a membrane bound space. Here Golgi apparatus and lysosomes and vacuoles are there, no? All these are all membrane bound cell organelles. Membrane bound means definitely membrane is present around the particular cell organelle. So membrane bound space found in the cytoplasm. So definitely all these kind of cell organelles are present in the cytoplasm only, no? Next one, this particular vacuole contains water, sap and excretory products and other materials. Tanni will be there different types of sap apadana liquid, so edo one kind of liquid. Next one, excretory products means after completion of metabolic activities excretory materials formation takes place, no? So such kind of waste materials and other materials so which are not useful for the plant cell. Okay, what are the materials which are not useful or not utilized for the plant cell now? So such kind of waste materials all are stored in the vacuoles. Point you understood, so this is save panikonga. Next one, the vacuole is bound by single membrane called tonoplast. So suppose one spherical shaped ball is a beans ball mele rubber will be there, no? That particular structure only. So here suppose that kind of structure if you see in vacuoles, so that particular membrane we will be calling as tonoplast. So simply what is the thing? The vacuole is bound by a single membrane called tonoplast. And what is the liquid present in the within the tonoplast? So we can be taken as tonoplasm. Understood? So means, so what is a single membrane bound cell organelle or single membrane bound structure is there which covers the vacuole, we are calling as tonoplast. And what is the liquid which is present within the tonoplast, we are calling as tonoplasm. That point through my voice you can get. Next information is next one, the plant cells. In plant cells, vacuole occupy up to 90% of its volume of the cell. Point you understood. So means plant cells once we took studied, no? Suppose if you take a plant cell or the matured plant cell there, large central vacuole is present, we seen in introduction. Remembered? So 3 points, chloroplast, mitochondria, large central vacuole and centrioles. So by taking of these 4 characteristics, no, we have studied the differences between the plant cell and the animal cell. If in plant cells, no, so centrioles will not be there, and in animal cells, no, chloroplast will not be there. So that kind of structure, I think so a few days back we have seen. Did you see that video? So later there we learned one point. What is that? In a plant cells, no, large central vacuole will be there. So that particular large central vacuole can occupy up to 90% of the volume of the cell. 90% of the volume of the cell definitely is occupied by the vacuole. That's why while writing no, we are calling as large central vacuole is present in a plant cells we are writing. Next one, ఇది important point రా, don't forget this one. So ఇది కొంచెం you cannot understand this one, but when you will come to the school no, at the time this particular point I will explain, but you have to remind to me. Right, now let us start to discuss. Now in plant cells or in plants, the tonoplast means a single membrane bound structure no, of vacuole adema. So this particular tonoplast facilitates the transport of a number of ions and other materials against the concentration gradient in the vacuole. Against the concentration gradient means, so here I told you where in a Golgi, like in cell membrane explanation I have given passive transport, active transport. Next one, concentration gradient, against the concentration gradient I told you no, concentration gradient means the movement or the transport of ions from the higher concentration to lower concentration. Against to concentration gradient means, once you see against concentration gradient means from low to high. So here when the food materials or the materials transportation is going on against the concentration gradient into the vacuole means definitely it requires the which one, energy in the form of ATP. So means there, suppose if the ions materials or the any kind of ions or the minerals movement is going on against the concentration gradient means definitely it requires energy help in the form of ATP. So thereby this particular point you try to remember. Okay ma. So hence their concentration is significantly higher in the vacuole than the cytoplasm. So thereby when food materials or the waste materials are going to be deposited in the vacuole, automatically what will be happen after somewhat time no, within the vacuole concentration of the materials will be increased. So thereby within the vacuole, so concentration of the materials, minerals, ions concentration will be more when compared to the cytoplasm. Next information is, so here in amoeba, contractile vacuole is important ma. So in amoeba, contractile vacuole is important for the osmo regulation. Osmo regulation means now we see now, that means regulation of which one movement of or the transport of ions. So how many number of ions must come inside and what are the ions must go outside. So means here this kind of regulation process we will be considering simply as osmo regulation. So means in amoeba, actually generally it will be there in aquatic conditions no, within in a tanninali will be there amoeba. So this particular amoeba consists of contractile vacuole. So what is the use of this contractile vacuole sir means helps in osmo regulation of ions or minerals, and one more is excretion. So what are the waste materials which are present in this cell no, definitely because of the presence of this contractile vacuole, so excretion process also takes place. Next one, in many cells, so for example in protist members, if you take protist members no, food vacuoles are present. So these vacuoles so these are all present. How these food materials formation takes place means, so by engulfing the food particles. So means here food vacuoles, so food vacuoles of formation takes place. How these food vacuoles of formation takes place means because of the taking. Engulfing means taking, means swallowing. Simply if you take there no correct word no, engulfing means takes or consume or takes or like a swallow. Okay, so if you take the such kind of means here because of the swallowing or engulfing of food materials in a protist members, which one formation takes place? Food vacuoles formation takes place. So now here in this video, so what are the main important points we have discussed in this side means in lysosomes information, hydrolytic enzymes are there, lipids, proteins, carbohydrates, and what are the hydrolytic enzymes are present in the lysosomes no, very active at acidic pH. This particular point definitely I will give in slip test examinations. Next on a vacuole point of view, so here the vacuole membrane, single membranous bound structure is there. So the vacuole membrane we will be calling as a tonoplast, and what is the sap which is present within the tonoplasm we are calling, and in plant cells no, vacuole occupy the volume of the cell, definitely 90% of the volume of the cell is occupied by the which one? Vacuole. Next one, so definitely this particular vacuole helps for the storage of excretory products, water, sap. Likewise there are different types of materials. Next one here, ipu definitely one more time once I'll explain. So here, what are the waste materials which are present in the cytoplasm? Definitely must move into the vacuole. How? Against the concentration gradient with the help of ATP or by consuming of ATP. Later in amoeba members we are observing the contractile vacuole, and this particular contractile vacuole helps in osmo regulation and excretion, and in a protist members food vacuoles are there. These are all formed by the engulfing of food materials. So this is a small information regarding endomembrane system. In a previous video we have discussed the endoplasmic reticulum. In this video we have covered remaining 3 cell organelles of endomembrane system, that is Golgi apparatus, lysosomes, and one more is vacuoles. So easy concept only, so try to watch this video carefully and gain the knowledge well. Thank you and be safe at home.